This video is about taste and smell, some special senses of human beings. Human beings, in the sense we are dealing with human and physiology. That's why our learning objectives include: list the types of taste and describe taste receptors, trace the taste pathways, trace the olfactory pathway, define disorders of taste and smell. So let's start. The senses of taste and smell allow us to appreciate the world around us and allows us to separate undesirable or even lethal foods from those that are pleasant to eat and nutritious. In addition, they elicit physiological responses that are involved in the digestion and utilization of foods. The sense of smell also allows animals to recognize the proximity of other animals and even individual animals. Finally, both senses are strongly tied to primitive emotional and behavioral functions of our nervous system. In this chapter, we discuss how taste and smell stimuli are detected and how they are encoded in neural signals transmitted to the brain. So let's start. Sense of taste. Taste is mainly a function of the taste buds in the mouth. But it is a common experience that one's sense of smell also contributes strongly to taste perception. Sense of smell is also very important. In addition, the texture of food, as detected by tactile senses of mouth, and the presence of substances in the food that stimulate pain endings, such as pepper, greatly alter the taste experience. The importance of taste lies in the fact that it allows a person to select food in accordance with desires and often in accord with the bodily tissues, metabolic need for specific substances. Primary sensation of taste. The identities of the specific chemicals that excite different taste receptors are all known, not well all known. Even so, psychophysiological and neurophysiological studies have identified at least 13 probable chemical receptors in the taste cells as follows: sodium receptors, two sodium receptors, two potassium receptors, one chloride receptor, one adenosine receptor, one eosin receptor, along with two sweet receptors and two bitter receptors, one glutamate receptor and one hydrogen ion receptors. Once again, which are they? Sodium receptors, potassium receptors, chloride receptors, adenosine receptors, eosin receptors, sweet receptors, bitter receptors, glutamate receptors, and hydrogen ion receptors. For practical analysis of taste, the aforementioned receptor capillaries have also been grouped into 5 general categories called the primary sensations of taste. They are salty, sweet, bitter, and umami taste. I'm not sure about the pronunciation of all this. A person can perceive hundreds of different tastes. They are all thought to be combinations of the elementary taste sensations, just as all of these colors we see are combinations of 3 primary colors. Sour taste. The sour taste is caused by acids, that is by the hydrogen ion concentration, and the intensity of this taste sensation is approximately proportional to the logarithm of the hydrogen ion concentration. More acidic the food, the stronger the sour sensation becomes. Then salty taste. Next, salty taste. The salty taste is elicited by ionized salts, mainly the sodium ion concentration. The quality of the taste varies somewhat between one salt to another because some of the salts elicit other taste sensations in addition to saltness. The cations of the salts, especially sodium cations, are mainly responsible for salty taste, but the anions also contribute to a lesser extent. So, sour taste is due to hydrogen ion concentration. Salty taste is due to sodium ion concentration. Then sweet taste is not caused by any single class of the sweet taste is not caused by any single class of chemicals. Some of the types of chemicals that cause this taste include sugars, glycols, alcohols, aldehydes, ketones, amides, esters, some amino acids, and some small proteins. Sulfonic acids, halogenated acids, and inorganic salts of lead and beryllium. Sulfonic acids, halogenated acids, and inorganic salts of lead and beryllium. Not specifically that most of the substances that cause the sweet taste are organic chemicals to a greater extent. Organic. It is especially interesting that slight changes in the chemical nature, such as addition of a simple radical, can often change the substance from a sweet to bitter. Bitter taste. Next, bitter taste. The bitter taste, like the sweet taste, is not caused by any single type of chemical agent. Here again, the substances that give the bitter taste are almost entirely organic substances. They are of organic origin. Two particular classes of substances are especially likely to cause bitter taste sensations, like long chain organic substances that contain nitrogen, and alkaloids. The alkaloids include many of the drugs used in medications such as quinine, caffeine, nicotine, and strychnine, etc. Bitter taste is caused by long chain organic substances that contain nitrogen and alkaloids. Some substances that initially taste have a bitter aftertaste. This characteristic too is true in saccharin, which makes the substance objectionable to some people. The bitter taste, when it occurs in high intensity, usually causes the person or animal to reject the food. This reaction is undoubtedly an important function of a bitter taste sensation because many deadly toxins found in poisonous plants are alkaloids. Many deadly toxins found in poisonous plants are alkaloids, and they, to a great extent, cause the bitter taste. So, all of these alkaloids cause an intensely bitter taste, usually followed by the rejection of food. Then, umami taste. Umami is a Japanese word meaning delicious, designate a pleasant taste sensation that qualitatively different from salty, sweet, or bitter taste. Umami is the dominant taste of food containing L-glutamate. L-glutamate. L-glutamate, such as meat extracts and aging cheese, and some physiologists consider it to be a separate fifth category of primary taste stimuli. A taste receptor for L-glutamate, just remember this name, may be related to one of the glutamate receptors that are also expressed in neuronal synapses of the brain. However, the precise molecular mechanism responsible for the umami taste is still unclear. Then threshold for taste. The threshold for stimulation of the sour taste by hydrochloric acid average 0.0009 N. For stimulation of the salty taste by sodium chloride 0.01 M. Taste blindness. Some people are taste blind for certain substances, especially for different types of thiourea compounds, a substance used frequently by psychologists for demonstrating taste blindness is PTC paper, which we might have tested, like phenyl thiocarbamide. Phenyl thiocarbamide. Phenyl thiocarbamide, for which about 15 to 30% of all people exhibit taste blindness. The exact percentage depends on the method of testing and the concentration of the substance. Then taste bud and its function. Taste bud is of a diameter of about 1 by 30th mm and a length of about 1 by 16th mm. Taste bud and its function. We just learned the diameter and length. The taste bud is composed of about 50 modified epithelial cells. 50 modified epithelial cells, some of which are supporting cells called sustentacular cells, and others of which are taste cells. Sustentacular cells and taste cells. Taste cells are continuously being replaced by mitotic division of surrounding epithelial cells. So, some taste buds are young cells. Others are mature cells that lie toward the center of the bud. These soon break off and dissolve. The lifespan of each taste bud is about 10 days in lower mammals, but it is unknown for humans. Stratified squamous epithelium is there. Microvilli, a pore will be there. Then nerve fibers, taste cells, and subepithelial connective tissue. These are the major regions of a taste bud. The outer tips of the taste cells are the outer tips of taste cells are arranged around a minute taste pore. From the tip of each taste cell, several microvilli or taste hairs protrude outward into the taste pore to approach the cavity of the mouth. This microvilli provide the receptor surface for taste. Interwoven around the bodies of this taste cell is a branching terminal network of taste nerve fibers that are stimulated by the taste receptor cells. Some of these fibers invaginate into the folds of the taste bud cell membrane. Many vesicles form beneath the cell membrane near the fibers. These are believed to be these vesicles contain the neurotransmitter substance that is released during the cell membrane to excite the nerve fiber endings in response to taste stimulation. And location of the taste buds. The taste buds are found on three types of papillae on the tongue as follows. A large number of taste buds are on the walls of tufts that surround the circumvallate papillae. Tufts that surround the circumvallate papillae, which form the V-line on the surface of the posterior tongue, and moderate numbers of true tube buds, taste buds are in the fungiform papillae over the flat anterior surface of the tongue, and moderate numbers are on the foliate papillae located in the folds along the lateral surface of the tongue. Then additional taste buds are located on the palate, and a few are found in the tonsillar pillars. Once again, circumvallate papillae, V-line in the surface of posterior tongue, moderate numbers on the taste buds under the fungiform papillae, then some on the tongue, surface of the tongue, and moderate numbers are on the foliate papillae, some on the tonsillar pillars and some on epiglottis, and even in the proximal esophagus. Adults have about 300 to 10,000, 3,000 to 10,000 taste buds, and children have a few more. Children have a few more in the sense they are able to, detect some more tastes, I guess. Then beyond the age of 45 years, many taste buds degenerate, causing taste sensitivity to decrease in old age. Taste sensitivity to decrease in old age. Specificity of taste buds for a primary taste stimulus. Microelectrode studies from single taste bud show that each taste bud usually responds mostly to one of the 5 primary taste stimuli and the taste substances are at low concentration. However, at high concentration, most buds can be excited by 2 or more of the primary taste stimuli as well as by few other taste stimulus that do not fit into the primary categories. Mechanism of stimulation of taste buds. Receptor potential. The membranes of the taste cell, like that of most other sensory receptor cells, is negatively charged on the inside with respect to the outside. Negatively charged on the inside. Application of a taste substance to the taste hairs causes partial loss of this negative potential, that is, the taste cell becomes depolarized. In most instances, the decrease in potential within a wide range is approximately proportional to the logarithm of concentration of the stimulating substance. The change in the electrical potential of the taste cell is called the receptor potential for taste. Electrical potential change. The mechanism by which most stimulating substances react with the taste villi to initiate the receptor potential is by binding of the taste chemical to a protein receptor molecule that lies on the outer surface of the taste receptor cell near to or protruding through a villous membrane. In action, in turn, opens ion channels which allows positively charged sodium ions or hydrogen ions to enter and depolarize the normal negativity of the cell. And the taste chemical is gradually washed away from the taste villus by a saliva. This removes the stimulus. The type of receptor protein in each taste villus determines the type of taste that will be perceived. The mechanism by which most stimulating substances react with the taste villi to initiate the receptor potential is by binding of the taste chemical to a protein receptor molecule that lies on the outer surface of the taste receptor cell near to the protruding through a villus membrane. In action, in turn, opens ion channels which allows positively charged sodium ions or hydrogen ions to enter and depolarize the normal negativity of the cell. Then the taste chemical is gradually washed away from the taste vellus by the saliva, which removes the stimulus. The type of receptor protein in each taste vellus determines the type of taste that will be perceived. For sodium ions and hydrogen ions, which elicit salty and sour taste sensations, respectively, the receptor protein opens specific ion channels on the apical membranes of the taste cells, thereby activating the receptors. However, for the sweet and bitter taste sensations, the proportions of the receptor protein molecule that protrude through the apical membrane act as second messenger transmitter substances inside the taste cells, and the second messengers cause intracellular chemical changes that elicited the taste signal. Generation of nerve impulses by the taste bud. Generation of nerve impulses by the taste bud. On first application of the taste stimulus, the rate of discharge of nerve fibers from the taste buds rises to a peak in a small fraction of a second, but then adapts within the next few seconds back to normal. Steady level as long as the taste stimulus remains. Thus, a strong intermediate signal is transmitted by the taste nerve, and a weaker continuous signal is transmitted as long as the taste bud is exposed to the taste stimulus. Transmission of the taste signal into the central nervous system. Taste impulses from the anterior two-thirds of the tongue pass first into lingual nerve, then through the chorda tympani into the facial nerve, and finally into the tractus solitarius. Once again, lingual nerve, then chorda tympani, then facial nerve, and then tractus solitarius in the brainstem. Taste sensations from the circumvallate papillae on the back of the tongue and from other posterior regions of the mouth and throat are transmitted through the glossopharyngeal nerve, also into the tractus solitarius, but a slightly more posterior level. Finally, a few taste signals are transmitted into the tractus solitarius from the base of the tongue and other parts of the pharyngeal region by way of a vagus nerve. Then all taste fibers synapse in the posterior brainstem in the nuclei of the tractus solitarius. This nucleus and second order neurons to small area in the ventral posterior medial nucleus. Ventral posterior medial nucleus of the thalamus, located slightly medial to the thalamic terminations of the facial region of the dorsal column medial levansal system. From the thalamus, third order neurons are transmitted to the lower tip of postcentral gyrus. Lower tip of postcentral gyrus in the parietal cerebral cortex. And this curls deep into the sylvian fissure into the adjacent orbicular insular area. This area lies slightly lateral, ventral, and rostral to the area for tongue tactile signals in cerebral somatic area 1. From this description of the taste pathways, it is evident that they closely parallel the somatosensory pathways from the tongue. Which are the regions? Just one quick recap. Lingual nerve, chorda tympani, facial nerve, tractus solitarius, glossopharyngeal nerve, brainstem, then tractus solitarius, and once again then tractus solitarius into vagus nerve, then ventral posterior medial nucleus, thalamus, then lower tip of the postcentral gyrus in the posterior cerebral cortex, along with sylvian fissure and insular area. And taste reflexes are integrated in the brainstem. From the tractus solitarius we just mentioned, many taste signals are transmitted within the brainstem itself directly into the superior and inferior salivary nucleus. These areas transmit signals to the submandibular, sublingual, and parotid gland to help control the secretion of saliva during the digestion and ingestion of food. Rapid adaptation of taste. Everyone is familiar with the fact that taste sensations adapt rapidly, often almost completely within a minute or so to continuous stimulation. Yet from electrophysiological studies of taste nerve fibers, it is clear that adaptation to the taste buds themselves usually accounts for no more than about half of this rapid taste adaptation. Therefore, the final extreme degree of adaptation that occurs in the sensation of taste almost certainly occurs in the central nervous system. Mechanisms are not known. At any rate, it is a mechanism different from that of many other sensory systems which adapt almost entirely at the receptors. Disorders of taste. Disorders of taste are either those that involve a loss of ability to perceive taste or those linked to the distorted sensation of taste. Distorted sensation of taste. Loss of ability or distorted sensation of taste. Loss of ability of taste is called ageusia, and while the reduced ability to taste is called hypogeusia. Distorted one is called dysgeusia. Dysgeusia. These conditions can occur due to advanced age or medications and constitute an important threat to the quality of life. Next topic is about sense of smell. Smell is the least understood of our senses, partly because the sense of smell is a subjective phenomenon that cannot be studied with ease in lower animals. Another complicating problem is that the sense of smell is poorly developed in human beings compared with the sense of smell in many lower animals. Olfactory membrane. The olfactory membrane, the histology of which is very important, lies in the superior part of each nostril. Medially, the olfactory membrane folds downward along the surface of the superior septum and folds over the superior turbinate and even over a small portion of the upper surface of the middle turbinate. In each nostril, the olfactory membrane has a surface area of about 2.4 cm square. Olfactory cells are the receptor cells for smell sensation. The olfactory cells are actually bipolar nerve cells. Bipolar nerve cells derived originally from the central nervous system. There are about 100 millions of these cells in the olfactory epithelium, interspersed among sustentacular cells. The mucosal end of the olfactory cell forms a knob from which 4 to 25 olfactory hairs, also called olfactory cilia, measuring 0.3 μm in diameter and up to 200 μm in length, project into the mucus that coats the inner surface of the nasal cavity. These projecting olfactory cilia form a dense mat in the mucus and in the cilia that react to odors in the air and stimulate the olfactory cells. So, spaced among the olfactory cell and the olfactory membrane are many small Bowman glands, BOWMAN glands that secrete mucus into the surface of the olfactory membrane. Bowman glands. And stimulation of the olfactory cells, mechanism of excitation of the olfactory cells. The projection of the portion of each olfactory cell that responds to the olfactory chemical stimuli is the olfactory cilia. The odorant substance, upon coming in contact with olfactory membrane surface, first diffuses into the mucus that covers the cilia and then it binds with receptor proteins in the membrane of each cilia. Each receptor protein is actually a long molecule that threads its way through the membrane, also 7 times, folding inward and outward. The odorant binds with the portion of the receptor outward. The odorant binds with the portion of the receptor protein that folds into the outside. The inside of the folding protein, also called the G protein, itself is a combination of 3 subunits. Upon excitation of the receptor protein, an alpha subunit breaks away from the G protein to activate adenylyl cyclase, which is attached to the inside of the ciliary membrane near the receptor cell body. The activated cyclase in turn converts many molecules of intracellular adenosine triphosphate into the cyclic adenosine monophosphate. Finally, the CAMP activates another nearby membrane protein called the gated sodium ion channel, which opens its gate and allows large number of sodium ions to pour through the membrane into the receptor cell cytoplasm. Gated sodium ion channel. The sodium ion increase the electrical potential in the positive direction inside the cell membrane, thus exciting the olfactory neuron and transmitting action potentials into the central nervous system by way of olfactory nerve. The importance of this mechanism for activating olfactory nerves is that it greatly multiplies the excitatory effect of even the weakest odorant. To summarize, activation of the receptor protein by the odorant substance activates the G protein complex, which in turn activates multiple molecules of adenylyl cyclase inside the olfactory cell membrane, which causes the formation of many times more molecules of CAMP. And finally, the CAMP opens still many times more sodium ion channels. Therefore, even a minute concentration of a specific odorant initiates a cascading event that opens extremely large number of sodium channels. This process accounts for the exquisite sensitivity of odorant olfactory neurons to even the slightest amount of odorant. In addition to the basic chemical mechanism by which the olfactory cells are stimulated, several physical factors affect the degree of stimulation. First, only volatile substances that can be sniffed into the nostrils can be smelled. Second, the stimulating substance must be at least slightly water soluble so that it can pass through the mucus to reach the olfactory cilia. Third, it is helpful for the substance to be at least slightly lipid soluble, presumably because lipid constituents of the cilium are a weak barrier to non-lipid soluble odorants. We tried odorant is there, odorant receptor, adenylyl cyclase, G protein, ATP, extracellular side, Na+, CMP, and cytoplasmic side. Membrane potentials and action potentials in olfactory cells. The membrane potential inside unstimulated olfactory cell is measured by microelectrode averaging about -55 MV. At this potential, most of the cells generate continuous action potentials at a very slow rate, varying from 1 every 20 seconds up to 2 to 3 per second. Most odorants cause depolarization of the olfactory cell membrane, decreasing the negative potential in the cell from the normal level of -55 to -30 MV or less, that is changing the voltage in the positive direction. Along with this, the number of action potentials increased to 20 to 30 second inverse, which is high rate for minute olfactory nerve fibers. Over a wide range, the rate of olfactory nerve impulses change approximately in proportion to the logarithm of the stimulus strength, which demonstrates that is olfactory receptors obey principles of transduction similar to those of other sensory receptors. Then rapid adaptation of olfactory sensation. The olfactory receptors adapt about 50% in the first second or so after stimulation. Thereafter, they adapt very little and very slowly. Yet we all know our own experience that smell sensations adapt almost to extinction within a minute or so after entering a strongly odorous atmosphere, because this psychological adaptation is far greater than the degree of adaptation to the receptors. It is almost certain that most of the additional adaptation occurs within the central nervous system, which seems to be true for the adaptation of taste sensation as well. The following neuronal mechanism for the adaptation is postulated. Large number of centrifugal nerve fibers pass from the olfactory regions of the brain backward among the olfactory tract and terminate on special inhibitory cells in the olfactory bulb, the granule cells. It has been postulated that after the onset of an olfactory stimulus, the central nervous system quickly develops strong feedback inhibition to suppress relay of the smell signals through the olfactory bulb. Search for the primary sensations of smell. In the past, most physiologists were convinced that the many smell sensations are served with very few discrete primary sensations, in the same way that vision and taste are served by the few select primary sensations. On the basis of psychological studies, one attempt to classify the sensations are as follows: camphorous, musky, floral, pepperminty, ethereal, pungent, and putrid. It is certain that this list does not represent the true primary sensations of smell. In recent years, multiple clues, including specific studies of the genes that encode for the receptor proteins, suggest the existence of at least 100 primary sensations of smell. 100 primary sensations of smell. A marked contrast of only 3 primary sensations of color detected by the eyes and only 5 primary sensations of taste detected by the tongue. Some studies suggest that there may be as many as 1,000 different types of odor and receptors. Further support for the many primary sensations of smell is that people have been found who have odor blindness for single substances. Odor blindness is also there. Such a discrete odor blindness has been identified for more than 50 different substances. It is presumed that odor blindness for each substance represents lack of the appropriate receptor protein in the olfactory cells for that particular substance. Then affective nature of smell. Smell, even more so than taste, has the affective quality of either pleasantness or unpleasantness, and this smell is probably even more important than taste for the selection of food. Indeed, a person who has previously eaten food that disgusted him, disagreed with him, or her is even nauseated by the smell of the same food on a second occasion. Conversely, perfume of a slight quality can be a powerful stimulant of human emotions. In addition, in some animals, odors are the primary excitement of sexual drive. Threshold for smell. One of the principal characteristics of smell is the minute quantity of stimulating agent in the air that can elicit a smell sensation. For instance, a substance, methyl mercaptan, can be smelled when only 1/25 trillionth of a germ is present in each milliliter of air. Because of this very low threshold, the substance is mixed with a natural gas to give the gas an odor that can be detected when even small amount of gas leak from a pipeline. Methyl mercaptan, not ethyl, methyl mercaptan. Sensations of smell signals into the central nervous system. The olfactory portions of the brain were among the first brain structures developed in primitive animals, and much of the remainder of the brain developed around these olfactory beginnings. In fact, part of the brain that originally subserved olfactory later evolved into the basal brain structures that control emotions and other aspects of human behavior. We call this system the limbic system. Transmission of olfactory signals into the olfactory bulb. The olfactory bulb is very important. The olfactory nerve fiber lead backward from the bulb is called radial nerve 1 or the olfactory tract. Radial nerve 1 or the olfactory tract. In reality, both the tract and the bulb area are anterior outgrowth of brain tissue from the base of the brain. The bulbous enlargement at its end, the olfactory bulb, lies in the cribriform plate separating the brain cavity from the upper reaches of the nasal cavity. The cribriform plate has multiple small perforations through which the equal volumes of small nerves pass upward from the olfactory membrane in the nasal cavity to enter the olfactory bulb in the cranial cavity. Close relation between the olfactory cell and the olfactory membrane and the olfactory bulb, showing short axons from the olfactory cell terminating in the multiple globular structures within the primary bulb called the glomeruli. Each bulb has separate 1,000 each glomeruli, each of which are terminus of about 25,000 axons from the olfactory cell. Each glomeruli also terminate for dendrites from about 25 large mitral cells and about small tufted cells, the cell bodies of which lie in the olfactory bulb receptor superior to the glomeruli. This dendrites receive synapses from the olfactory cell neurons, and the mitral and tufted cells send axons through the olfactory tract to transmit olfactory signals to highest levels of the central nervous system. Some researchers suggest that different glomeruli respond to different orders. It is different glomeruli respond to different orders. It is orders. So it is possible that specific glomeruli are the real clue for analysis of different order signals transmitted into the central nervous system. Primitive and newer olfactory pathways into the central nervous system. The olfactory tract enters the brain at the anterior junction between the mesencephalon and the cerebrum. The tract divides into two pathways, one passing medially into the medial olfactory area and the brainstem, and the other passing laterally into the lateral olfactory area. The medial olfactory area represents very primitive olfactory system, whereas the lateral olfactory area is input less old olfactory system and a newer system. The primitive olfactory system, the medial olfactory area. The medial olfactory area consists of a group of nuclei located in the mid-basal portions of the brain immediately anterior to the hypothalamus. Most conspicuous of the septal nuclei, which are midline nuclei that feed into the hypothalamus and other primitive portions of the brain's limbic system. This is the brain area most concerned with the basic behavior. The importance of this medial olfactory area is best understood by considering what happens in animals when the lateral olfactory areas in both sides of the brain are removed and only the medial system remains. The removal of these areas hardly affects the more primitive response of olfaction, such as licking the brain, salivation, and other feeling responses caused by the smell of the food or by basic emotional drives associated with smell. The lateral olfactory area is composed mainly of the prepiriform and piriform cortex plus the cortical portion of the amygdaloid nuclei. From these areas, signal pathways pass into almost all portions of the limbic system, especially into primitive portions such as hippocampus, which seem to be more important for learning to like or dislike certain foods depending on one's experience with them. For instance, it is believed that lateral olfactory area is many connections with the limbic behavioral system cause a person to develop an absolute aversion to foods that cause nausea and vomiting. An important feature of the lateral olfactory area is many signal pathways from this area also feed directly into an older part of the cerebral cortex called the paleocortex in the anterior medial portion of the temporal lobe. Paleocortex in the anterior medial portion of the temporal lobe. This area is the only area of the entire cerebral cortex where sensory signals pass directly by the cortex without passing first to the thalamus. A newer olfactory pathway that passes through the thalamus, passing to the dorsomedial thalamic nucleus and then passing to the lateroposterior quadrant of the orbitofrontal cortex has been found. On the basis of studies on monkeys, this newer system probably helps in the conscious analysis of food. This appeared to be a primitive olfactory system that subserves the basic olfactory reflexes, and older system, a less old system, provide automatic but partially learned control of food intake and aversion of toxic and unhealthy foods, and a newer system that is comparable to most of the other cortical sensory systems and is used for subconscious perception and analysis of olfaction. Disorders of smell, like those of taste, are either those that involve a loss of ability to smell or those linked to distorted sensation of smell. Loss of ability of smell is called anosmia. Anosmia. Remember like that. While a reduced ability to taste is called to smell is called hyposmia. A distorted sense of smell is called dysomia or parosmia, and particularly when an unpleasant perception of an odor is present. Disorders of smell and taste go hand in hand in the perception of a patient. This is not surprising since both taste and smell are involved in the perception of flavor. Thank you for listening.